This is Professor Doherty, and today we're going to be covering Chapter 3 in Human Physiology, Cells and Tissues. So this is basically cell biology. Let's take a look at what we're going to be discussing today. So today what we're going to do is we're going to finish up on biomolecules and looking at the structure of nucleotides. It's our final biomolecule. And then we'll move into looking at the difference between ions, isotopes, and then we'll talk about functional groups which can play a big role in chemistry. And then we'll look at cell membranes and how they can control the movement of molecules through themselves, as well as some important factors like pH, which have a huge impact on the chemistry in your body, energy and chemical reactions, and then finally enzymes. So a variety of topics to talk about today. This just shows the diversity of cell biology, really. Okay, so to begin with, we'll need to look at the structure of nucleotides. So with that, I'm going to flip over to my whiteboard here. And so let's go ahead and take a look at our fourth biomolecule. So remember that nucleotides are the fourth biomolecule. And if we look at nucleotides, they consist of several components to them. The first part is a sugar ring. So I'm going to start down here with one of our sugar components. And again, you should draw anything that I draw. Here I'm going to draw a ribose molecule. And so this is a ribose, and we know that because it has the oxygen down here at the second carbon. Now coming off the ribose, and I'll label all these components later, is a phosphate group that's going to come off of the carbon that hangs off of the ribose up here. And so I'm going to fully draw out this phosphate group. And then on the other side over here, coming off this carbon, I'm going to draw a very long connection here just to keep it out of the way of the rest of this picture. But here we're going to have what's called a nitrogenous base, and that's going to have nitrogen in it. And so, but we'll still have our ring structure here. So I am going to draw adenine here. And then there's some double bonds here too. Okay. So let's talk about the components of this. So this is a generic nucleotide. Well, this is actually a specific nucleotide that I've depicted here, but with the generic structure. This is adenine that I've drawn here. Now, out of the nucleotides, you may recall that there are 5 nucleotides, so let's go ahead and list those real quick. Those are adenine, guanine, cytosine, thymine, and the last one that's used only in RNA is uracil. Okay. These all have similar structure with the exception of their nitrogenous base. So let's go ahead and lay those out. So this part down here is a sugar ring. And the sugar is either ribose or deoxyribose. Oops. It can be either one. In this case, I've drawn ribose because there is an oxygen down here off of this carbon. Second, like I mentioned up over here, this portion of this drawing is a phosphate group. And then lastly up here, this is the variable part. Okay, so all nucleotides have a phosphate group and a sugar group. They also have a nucleotide, but the nucleotide is what changes the most. And so this is called a nitrogenous base. And I should say it's the nitrogenous base that changes the most. You can have either a double ring structure up here like I've drawn in adenine, or a single ring structure up here. All of them, the reason they're called nitrogenous bases is because they have a lot of nitrogen in them, as you can see here. You can also see this is the first time I've drawn a ring structure that has a different corner molecule rather than a carbon, and this is how you would draw that. So basically each of these corners, remember, has a carbon, and then we have nitrogen in 4 of the corners here on these rings, and then 1 extra nitrogen group off the top, the amine group. Now, depending on the nitrogenous base will determine which of the nucleotides this actually is. So again, all the nucleotides have a sugar and a phosphate, and then they change their nitrogenous base to become the individual nucleotides. Now, besides the nitrogenous bases being able to be exchanged out, you can also exchange out the sugars, as I've shown here. They can either be ribose or deoxyribose, but only those two. And they're either used to form RNA or to form DNA. So RNA in the case of ribose and DNA in the case of deoxyribose. All of these molecules are used as informational ways to transfer information. They can also be used, so nucleotides can also have secondary functions as well. They can also be used as energy transport vehicles. So for instance, you've heard of ATP, which is adenosine triphosphate. That essentially would be this molecule plus 2 additional phosphate groups added onto it. So ATP, ADP, NADH, you may have heard of some of these molecules. These are all energy transfer molecules, FADH2. Okay, and we'll talk about those more in later units. All right, so let's take a look at some of those back on our slide set. So you can see here is a list of single nucleotide molecules that can be used for energy transfer. So you should familiarize yourself with this list here. You can see that there are many things that I wrote down as well. ATP, ADP. Just understand that NAD and FAD are the same as NADH and FADH, just whether they're loaded or unloaded with high energy electrons. All right, so those are your 4 energy transport or energy transfer molecules, and then there is one that's also used for cell-to-cell communication, and that's called cyclic AMP or CAMP. All right, so now let's talk about functional groups. Functional groups are basically groups of molecules that move together around larger structural molecules. They move as a single unit. These functional groups are important because they can change the chemistry and biochemistry of a molecule. So let's go ahead and take a look at some of these. Go back to the drawing board here. And so let's go ahead and take a look now at functional groups. And some of these I've gone over in the past. You should write these down just so you know though. So amine or amino group. This has a structure of NH2 as you've seen, and you can draw it like this, or an alternative way to draw this would be, I'm going to draw it slightly up here just so it's out of the way, is in its individual molecule form like this with the bonds between them shown. This is also NH2. Okay. Next is a carboxyl group. Carboxyl group can be denoted as COOH. It also has a secondary denotation as COO minus if you take the hydrogen off of it. Or you can draw it in its long form like this. All right. Now you can also take another long form of this would be like this with a negative out over here. So I'll do that negative in red so it shows separately. And here we've just taken the hydrogen off. The reason why we draw it sometimes like this with the hydrogen off is because hydrogen has a tendency to easily pop off of these carboxyl groups. That's one of the reasons why they're often called acids. Next is a hydroxyl group. A hydroxyl group looks like this, just simple OH, or it can be drawn in a longer format like this. Typically it's rare that you see it like this though. And then the final one is a phosphate group, which of course I've drawn several different times. However, you can also write it like this, excuse me, H2PO4, or just as PO4 2 minus. Okay, these are the equivalent. Again, here we have the hydrogens popping off, and so in long format that would look like this, which I know I've drawn several times before. Or if you take the hydrogens off, you can also draw it like this. And here again I'm going to mark these as negative charged oxygen molecules because they're going to have a tendency to keep the electron from the hydrogen. So then the hydrogen pops off, okay? Or in this case, two hydrogens pop off. These molecules that have the electronegativity on them are usually polar molecules, and so you'll find those listed as polar molecules, and you'll see them on polar portions of other molecules. Now these functional groups are important because as they move around on other long chain molecules, they can affect the chemistry of those molecules. As you can see here, they can make them, for instance, somewhat polar when they were, if they're, for instance, if they're stuck to a non-polar molecule, they can make that molecule somewhat polar, and so that can change the chemistry of that molecule. Okay, let's go ahead and go back to our slide set. So next I want to review ions. So once again, I want you to understand that ions are atoms or molecules that have a charge on them. And so here are some of the important ions of your body. These are very important. Many of these are. So you will get to know these. You should get to know them now, but you will get to know these over time throughout the course of this class. Sodium is an ion. It has a positive charge on it. Potassium is an ion. It has a positive charge. So is calcium. It actually has a double positive charge. And hydrogen. These are all extremely important. Magnesium I don't get into as much in this class. Some important anions include bicarbonate, chloride we see quite a bit, and phosphate. Not so much. I don't talk so much about the sulfate. Now, two other terms that you should understand here are cations and anions. These denote either positively charged molecules in the case of cations, or anions being negatively charged. So if I ever say anion, you should understand I'm talking about a negatively charged molecule, usually an atom. As you can see here, many of these are atoms, but some of them are full molecules as well. Okay, ions are different from isotopes, and so I want you to understand the difference. We don't really deal with isotopes much in this class. I'm going to talk about them because people get them confused. Isotopes are important for medical, what's the term I'm looking for? Medical equipment. So for instance, many imaging pieces of equipment will look for isotopes and identify isotopes. So that is where they're more important. For instance, if you go into the field of like magnetic imaging, then you would be looking at isotopes. Okay, so let's take a look at what isotopes are. An isotope is when a molecule, or excuse me, when an atom gains or loses a neutron. So you can see here we'll start over here, we have a normal hydrogen atom. If this normal hydrogen atom gains a neutron in its nucleus, and here you can see it's gained this little red guy, this causes it to become an isotope. In this case, you can call it just a hydrogen isotope. I believe this is deuterium, if I remember correctly. It actually has a name. The difference between an isotope and an ion is when we talk about an ion is when you're gaining or losing an electron. All right, so ions, you're gaining or losing an electron. Here you can see the hydrogen, normal hydrogen molecule over here, or atom over here, and then we've lost an electron. All right, so isotopes gain or lose neutrons, ions gain or lose electrons. You can see how ions are much more important to us because they are essentially what forms chemistry within your body. All right, so let's now move into cell membranes and what cell membranes do and what they are. So cell membranes have a number of functions, and we'll talk about those in a moment, but I just want to show you some pictures here of what cell membranes look like. You can see that the outer coating of all of these cells is a cell membrane, and they can have a variety of shapes. So the shape of the cell membrane is going to dictate the shape of the cell along with the underlying protein structure. And these cell membranes are incredibly important in physiology because they control what goes into and out of the cell, and that determines the function of the cell itself. So let's talk about some of the functions of the cell membrane itself. All right, so some of the functions include, first of all, the cell membranes helps to physically isolate the cell. And it isolates the cell from its external environment. And just so you're aware, the external environment is called the extracellular fluid. So for instance, if I go over here on this cell, this big yellow fatty cell, the extracellular fluid would be surrounding the cell. The intracellular fluid or cytoplasm is inside the cell. The cell membrane physically isolates the cell from its surrounding environment. The second thing it does is it regulates. And here it's going to regulate the exchange of nutrients and waste. So nutrients and waste products. So basically, this cell membrane can control what goes out of the cell and what comes into the cell. And this is vitally important for the functioning of the cell. So it can bring in, let's just talk about gases. It can bring in oxygen or allow oxygen in and allow carbon dioxide out. So that way cellular respiration takes place. The third thing that it can do, and so these first two many people recognize, right? But the third thing that the cell membrane can do is communicate. It is vital for communication. And what I mean by this is that the cell membrane actually has little proteins and carbohydrates sticking off of the edge of it that can be used as receptors. And so, for instance, hormones can come in and attach to those receptors, and thereby when they attach, they can trigger a cascade of changes within the cell. And this is what I mean by communication. So they can receive signals. They can actually also send signals by releasing things like hormones or other chemicals. So the cell membrane is actually vital for the communication of the cell with the outside world. And then the fourth thing that they do, which should also be obvious, is structural support. So they give the cell its shape. The underlying protein components definitely help with the shape of the cell. Now, the other thing that you should understand about cell membranes is what they're made up of. So their composition, the composition of a cell membrane includes a lot of lipids and some proteins as well. Now, the lipids also include cholesterol as well as phospholipids. So we have both actually. Now, in addition to that, the composition of the cell membrane is it's what's called a fluid mosaic, meaning that there are other things within the lipids, things like proteins within the lipids, and there's also some carbohydrates as well. So they're usually attached to something. So carbohydrates usually come in the form of what we call glycoproteins or glycolipids. These parts down here, these proteins, glycoproteins, and glycolipids, have a tendency to float around within the structure of the lipids. Okay, so the phospholipid membrane. That's why we call this a fluid mosaic. It acts kind of like a two-dimensional fluid, and those proteins and glycolipids and glycoproteins can float around in that two-dimensional membrane. Now, these proteins have different names, and so you may hear the term, for instance, integral protein or peripheral protein. So an integral protein is one that is embedded into the phospholipid membrane. All right. It oftentimes will span the phospholipid membrane, but not always, so that's an integral protein. We also have what are called peripheral proteins. And the peripheral proteins are ones that just sit on the edge of the phospholipid membrane. All right. Now let's take a look at the structure of the phospholipid membrane. This drawing right here actually shows a pretty good representation of what the phospholipid membrane looks like. And what I recommend that you do is draw a picture of one of these. So a phospholipid, in this case, in this drawing here, hang on a sec, there it goes, looks like this. It has a head and two tails off of it. And let me describe for you what this is showing. This is a representation of a phosphate group. And these two tails, if you recall, are fatty acids. Now, if you remember also, the phosphate group is polar, while the fatty acids are nonpolar. So these are lipids down here, the fatty acids. Because of this, it creates a molecule that one end, the polar end, likes to be around water. We call this being hydrophilic. So this end of the molecule is hydrophilic, meaning it likes water. The other end, so the fatty acid end, is called hydrophobic. And this means water hating. Sounds harsh, but you can think of it as water disliking. It does not like to be around water. And so because of this, it gives this molecule kind of some unique properties. And you can see down here the way that this molecule will arrange itself is shown. I'll get my pointer back here. It's shown here. It will arrange itself into a double layer with the heads of the molecules out here pointing out towards where all the water fluid is on the outside of the cell. Now on the other side you get another layer of these phospholipids lining up in the opposite direction, and they're facing the water that is inside of the cell. So if we look, for instance, on the cell here, right, you can see that this dark line here would represent a layer of phospholipids, the cell membrane. Remember that the extracellular fluid outside of the cell is primarily water, and the cytosol inside of the cell is also primarily water. So you can see how these phospholipids line up in such a way that the phosphate heads are interacting with the water on the outside of the cell and the inside of the cell. They do that because they're hydrophilic, they like water, and the hydrophobic parts, the fatty acid chains, line up on the inside. Okay, so they're away from the water, so they don't have to be near the water. Now, this plays an important role in what can penetrate through the cell membrane. So what can penetrate through the cell membrane, and you should write this down, includes things like fatty molecules like steroids. Small molecules can get through this as well. And it eliminates or stops things from getting through also. The things that it blocks are charged molecules and large molecules. Okay, so block by size and by charge. Okay. Now, this is not true for every molecule. So some molecules may be small and they may have a tendency to penetrate, but they might be charged and that keeps them from penetrating. So there can be dichotomies as far as what can penetrate and what can't. But for the most part, fatty molecules can penetrate, polar molecules cannot. Okay, that's a good basis. And then also large molecules have a difficult time getting through these cell membranes. If we look at a more detailed picture of cell membranes shown here, you can see that they're quite complex. So not only are we seeing the phospholipid balls and chains here shown here in purple, but we're also seeing some other things that are integrated into the cell membrane. So for instance, these yellow molecules shown here and blown up here, these are cholesterol molecules. So in fact, the fatty area in the center of the cell membranes is even made fattier by these cholesterol that are added in here. So this makes basically a fatty layer in the middle here, which is a lot of what can go through the cell membrane. Now also we have these proteins that are shown in green here that are also integrated into the cell membrane, as well as some of these glycoproteins where you can see these glycogen chains shown like this that are coming off of a protein. So they're stuck to it. You can also see here some glycogen chains that look like they're stuck directly into the cell membrane. In fact, what they're attached to is a fatty molecule that would be embedded in the membrane. That would make this a glycolipid, in fact. Now, you can also see that some of these proteins, what they do is they actually form pores. So this is a more detailed picture of what these proteins look like. They oftentimes have this kind of wraparound look to themselves. And what they'll do is they'll make these kind of spinning cores here, and in the middle of this will be a hole. So in fact, in the center of this protein, it will actually have a channel that can allow through certain types of solutes or maybe even the solvent itself, which is water. And so in this way, we can actually move molecules through the cell membrane that normally would not be able to get through the cell membrane. And this becomes very important later on, and so we'll talk about this later on in the semester. This is just a general drawing of a cell membrane, and here you can see the cell membrane and how it separates the inner cytoplasm or intracellular fluid from the extracellular fluid that's around the rest of the cell. And then it also shows a number of the organelles and pieces that are actually inside of a typical cell. There's nothing really here that's super important that we haven't already gone over. Next, let's talk about pH. pH is a measure of the concentration of free hydrogen within a solution. All right, so pH, once again, is a measure of the concentration of free hydrogen in a solution. So as you add more hydrogen to a solution, it becomes more acidic, but that lowers the pH. And as you take out hydrogen, that makes it less acidic, and that actually raises the number of the pH. So that can be a little confusing for people on the pH scale. So you should understand some other concepts here, that an acid contributes hydrogen to a solution. So remember when I talked about like the carboxyl group COOH, and I said that the H had a tendency to pop off or come off of that molecule? That's why it's considered to be slightly acidic. So it's a mild acid, because that hydrogen will pop off and go into the solution, and that contributes to the acidity of the solution. Now on the other side of that is a base, and a base will reduce the amount of hydrogen in the solution, so it will absorb some of that hydrogen. A buffer is something that moderates changes in pH. So buffers, oftentimes a buffer has two components to it, a part that is slightly an acid and a part that is weakly a base, so a weak acid and a weak base. And what will happen is as more hydrogen is added into a solution, the buffer will actually take up that hydrogen. And as more hydrogen is taken out of a solution, the buffer will add more hydrogen to the solution. So it offsets either acids or bases that are being added to that particular solution. So that can be kind of complicated for those of you not familiar with it, so you might want to study up on that a little bit. Bicarbonate is the most important buffer in the human body, so shown here HCO3 minus. And this is a buffer primarily found in blood. That's where it really does most of its work. So, but you should become familiar with bicarbonate. We'll be talking about it a lot throughout the semester. And the reason why I'm focusing here on pH and why it's an important concept for physiology is because it has a tendency to change the shape of a molecule. So it can, for instance, denature proteins, which means unravel proteins. And proteins are one of our main molecules that does work within our body. And so, proteins are essentially enzymes, and enzymes do all sorts of functions for us. So if the pH in your body gets too far out of whack, it can basically destroy the functionality of these enzymes, as well as, transport molecules or proteins. Proteins do a ton of things. So pH can deeply affect those. Here is the pH scale. So a few things that you should know about the pH scale. The pH scale is actually a logarithmic scale. And what that means is that for every one number unit that you move, you're actually moving up tenfold in the amount of hydrogen that's in the solution. So if we look at the scale here, 7 is a neutral solution on this scale. As you go down, you become more acidic. So something that's a pH of 0 is very acidic, meaning that it has a lot of hydrogen ions in it. You can see down here towards the low end of the scale, excuse me, includes stomach acid, lemon juice, right? As we go up on the scale, we become more basic or more alkaline. Those are the same thing, basic and alkaline. And you can see up here at the top of the scale, 14 means that we have very little hydrogen within the solution. So sodium hydroxide here, for instance, a 1 molar solution of sodium hydroxide is very basic. Hair removers are very basic. And so you can see the scale here. You should become familiar with this scale as well as the formula for this scale. So let me pull this up. So the pH formula that you should know looks like this. So you should write this down. pH is equal to the negative log of the concentration of hydrogen ions within that solution. These brackets mean the concentration of. This is a formula that you should commit to memory. All right. So pH is equal to the negative log of the concentration of hydrogen in a solution. Another way to write this is to say that pH is equal to the log of 1 over the concentration of hydrogen. All right, so those are equivalent basically. So if you remove the negative sign from one of them, then you have to flip the hydrogen over here. Now, this second equation actually shows more nicely how the level of hydrogen will cause the number of on the pH scale to go down. So for instance, if the concentration of hydrogen here gets larger, it makes this overall number smaller, and that's going to drive this number down the scale. So as we add more hydrogen, it's going to move this number lower. And so that can be a confusing concept to people, right? The more hydrogen we have, the smaller the pH number. So the higher the concentration, the lower the pH, and vice versa. So definitely familiarize yourself with that. Okay, so that's pH. Like I said, 7 is neutral. And so now let's take a look at some energy and chemical reactions that are based on that energy. Again, these are other important concepts for the basis of physiology. I do like showing this picture here. This picture is a picture of just some of the biochemical reactions that take place in your body. And I just want to show you this just to show you how complex physiology is. It's an incredibly complex science. And some people will spend their entire lives, like PhDs will spend their lives just studying one part of this overall map of the physiology and biochemistry that takes place within your body. And this is not a complete map even, right? This is just part of it. So very complex. We're only studying a portion of this overall system, a very small portion of it, an overview of it. So I just want you to see how much more there is to physiology than what you may realize. Some of you guys might recognize this here. This is actually the Krebs cycle shown on that picture. So it's also shown here, right? This is a blow up of it. You can see all the steps in the Krebs cycle. You can also see, for those of you who have learned the Krebs cycle, that in fact the Krebs cycle is much more complex than what you probably learned. You probably just memorized the outside words are going around this circle, and maybe what energy molecules are produced. But you can see that there's a ton more stuff in here that you actually could spend a whole lifetime studying. So all right, so let's take a look at the energy that drives these reactions. So a few things to know. These are basically laws of physics that apply to physiology. Energy, you should understand that you can't make it from scratch. It only changes form. This is the first law of thermodynamics. In fact, those of you who have studied physics, there are exceptions to this, but for us, this is what you want to know. You can't make energy, okay? And then the second law of thermodynamics is that basically everything moves from order to disorder. And we call this entropy. E-N-T-R-O-P-Y. Entropy is the movement of molecules or anything else from order to disorder. Now, these two laws basically govern what happens biochemically inside of your body. And so let's talk about the energy that it's actually going to be used for this. First of all, we have free energy. Sometimes you might hear this called Gibbs free energy. And oftentimes this is listed as a delta G. So let me go ahead and write that. So this free energy can be called Gibbs free energy. And sometimes it's listed as delta G. And what that means is that means a change in free energy. So G representing free energy here. This is typically organized and usable energy, is this free energy. All right, so basically in the form of, for our purposes, something like a complex molecule, for instance, like glucose, would be a form of free energy that we can use because it's an organized system of energy. And we'll take a look at that more later. Entropy, like I mentioned, is disorder. And there's less work that's possible in a disordered system. Everything in the universe is moving towards disorder eventually. And there's always a ratio between these two systems, basically. So you have basically order versus disorder. So order, where there's energy stored in the order, or disorder or entropy. This leads to a couple of reaction types that I want you to know. The first is an exergonic reaction. So the term is shown up here, so you should understand this. An exergonic reaction is a reaction that releases energy. So the free energy in an exergonic reaction is negative. All right. So our Gibbs free energy in an exergonic reaction is negative. So this would involve a negative delta G. There's a release of energy. These reactions, exergonic reactions, are typically spontaneous. And a spontaneous reaction is a reaction that favors the formation of products at the conditions under which the reaction is occurring. Okay, so as you can see here, what we're taking a look at in this picture here is a ball rolling down a hill. So this is basically the example. So you can imagine that a ball that's on a hill has energy stored in it because it's been pushed up the hill, right? So there's potential energy stored in the ball. When it's released, that energy is released, and basically there's a loss of energy. And you can see this here too. The reason why I'm using this ball and hill as an example is because the same thing happens in your body. You can have reactants that can spontaneously want to move to become products, and there's a release of energy over the course of this slope here. So the reactants become products quite easily. Now, the other type of reaction that we're going to look at are endergonic reactions. With endergonic reactions, there has to be an input of energy. So if there's an input of energy, we would call this a positive delta G, or just regular delta G. Oftentimes you don't put the plus out in front of it, right? It's just assumed to be positive. So endergonic reactions have a positive delta G, meaning that energy has to be put into the system. So the plus sign here indicates that we have energy coming into the system. You can see that here with our example. Here we have a ball. If we push it up a hill, this requires energy for us to push this ball up this hill. So we're storing energy as potential energy inside the ball. The same thing can happen here if we, in our bodies, when we talk about reactions, we can take reactants and force them to become products by adding energy. So this type of a reaction would be an endergonic reaction. Typically what this involves is the reactants are usually simple molecules, and then the products at the end are more complex. Okay, so we're taking a bunch of simple molecules, like let's say simple carbons, right? And if we put them together in a chain to form a glucose ring, that takes energy, and so then we get glucose as our product. This is what plants do, right, with photosynthesis. They take carbon dioxide, which would be their simple reactants, and they take those carbon dioxide and they turn them into glucose, and that's their product, and they're storing energy in the form of glucose. Now up here, an exergonic reaction where we're losing energy would be, for instance, if we took glucose, if that was our reactant, and then we broke glucose down into its products of, let's say, carbon dioxide, like we do in our body, where we take glucose and turn it into carbon dioxide, and we pull energy out of that. We're taking a complex molecule and turning it into simpler molecules. Basically, we're moving towards entropy. Remember, entropy is we're moving from complex to simple. All right, here's another example of this. So imagine that we have a high diver here, right? He's on his diving board. He has stored energy right now, right? So he has a higher free energy, but it's not as stable. When he jumps off, he releases all that energy and then eventually moves to a lower energy state when he enters the water and gets to completion down here. This example is just another example of what happens in our body. So for instance, if we have molecules, diffusion is also a form of moving from a high energy state into a low energy state. If we have a bunch of molecules that are all crammed together in the corner of this container and we allow them to move around, eventually they will diffuse and move into a more chaotic state, a state of entropy here. So there's more energy in the system up here that's organized and less energy down here. Same thing with molecules like I was talking about. If we have a very complex molecule, like imagine that this is maybe glucose, it is going to have a higher energy state because it's more complex. If we break it apart, we're releasing energy from this molecule and causing it to become these smaller molecules. This is a lower energy state. This is basically the basics of how your body works in terms of breaking down molecules to get energy and then building up molecules that takes energy. Now, the transfer of this energy takes place through a molecule called ATP primarily. This is adenosine triphosphate. It has high energy bonds between the phosphate molecules, meaning that there are high energy electrons that are stored here. The formation of these bonds is endergonic, meaning that we have to put in the energy to build ATP. So if you think of this as the primary way, let me take a look here. If you think of this formation of bonds means that, in our case at least, we're going from ADP, which is a lower energy state molecule. We're putting in energy to make ATP. So here there has to be energy input. Okay. So in order to form this last phosphate bond, we have to input energy. Now when we break this, when we take ATP and move it into ADP, and have ATP become ADP, this darn thing, sorry. What happens is we get ATP then becoming ADP again. This is an exergonic reaction, and so we're breaking bonds, and here we're going to release energy. And that's basically what happens as you move energy around, is you make bonds and break them, adding energy where you're forming those bonds and then taking away energy where you're breaking those bonds. This is called a transfer between catabolic and anabolic pathways. So let me describe those pathways. Catabolic pathways are pathways where you break down molecules. So for instance, ATP to ADP over here, so taking a more complex molecule of ATP, breaking it into ADP, that would be considered a catabolic pathway. We're making a complex molecule less complex. An anabolic pathway is where you're making something more complex, you're adding energy. So here we're taking ADP and turning it into ATP, a more complex molecule. This up here would be a anabolic pathway. Now you may have heard this term before, I'm sure, in the form of anabolic steroids, right? So anabolic in that case is talking about the formation of muscle. So a more complex molecule than just the regular protein or amino acids that you started with. This is what an ATP molecule actually looks like. And so you can see here, here's the sugar base, right? Here is the adenine nitrogenous base that we've drawn before, and here are the 3 phosphate groups, 1, 2, and 3 here. So you can see that this is pretty much a type of nucleotide, right? So everything from here is the adenine nucleotide, and then we just added 2 phosphate molecules onto it. Now the bonds between these phosphate molecules, both here and here, contain energy that can be drawn off and used in other places. The highest amount of energy is actually in this third bond, which is why your body will go from ATP down to ADP and then back up to ATP again. So it'll go back and forth between these two, transferring energy. It typically doesn't like to cut off this one because there isn't as much energy in the bonds down here. Now here's another drawing of ATP. So again, here's the ribose sugar, here is adenine, and then here are the 3 phosphate groups out to the side. And through a process called hydrolysis, this is how we get energy out of ATP. So we're going to take off this 3rd phosphate group. And so here's another representation of that same molecule. You can see the 3 phosphate groups here, here, and here. In the process of hydrolysis, we add water. So here we have an enzyme that's going to add water to ATP. And when it does that, it breaks off this last phosphate group. It's called an inorganic phosphate and denoted as Pi, as you can see here. When this happens, remember that these are less complex molecules than this is, and so because of that we have a release of energy. All right. Now again, just to reiterate, the formation of bonds is endergonic, meaning that we have to add energy. The breaking of bonds is exergonic. So this down here is an exergonic reaction. We're losing energy. Now this energy can be used to do other work in other places of the body, and that's typically what we do. This next couple of slides here just shows how energy is transferred from catabolism over into cellular work. So in this case, catabolism here would be, let's say, eating food. So if you eat food in your digestive system, you're taking complex molecules in, like let's say sugar, so a glucose molecule, let's say. That is a complex molecule. You break that sugar apart, that releases energy. When you release that energy from the sugar molecule, what you do is you store it onto an ADP by putting an extra phosphate onto that ATP and forming ATP. When you then do work, so let's say you start working out and lifting weights, you're now taking that energy that's been stored on the ADP as ATP, and you break ATP apart. You release work for you to move your muscles, and that breaks ATP into ADP and inorganic phosphate, and then it goes through the entire cycle again and again and again. Now, what can we use this ATP for? I mean, there's a whole variety of uses for this ATP and the energy stored on it. So this is just showing a few things that we can do. We can do mechanical work, like I just mentioned, flexing muscles. In this case, what this picture is showing is a motor protein actually moving along a tubule pathway and carrying an organelle with it. This is going to require energy for it to move. We can also use it, this is a big place where we use it as well, membrane transport, and this will be a big focus of what we look at this semester. So we can move molecules from areas of high concentration or excuse me, areas of low concentration to high concentration using ATP, because that requires energy for us to do that. And so here you can see a protein pump that's doing this. And then another way that we can do this is we can do it, use this energy to perform chemical work, where here we're taking glutamic acid and ammonia and making glutamine out of it. This is a more complex molecule, so it requires us to input energy in order to get this reaction to go. And then the end product, of course, is that our ATP becomes ADP because it's given up some of its energy. Now, these reactions will result in what's called these delta G changes, so change in free energy. So for instance, here in this reaction of glutamate to become glutamine, we have to input energy because we're taking two smaller parts and making them form a more complex molecule here. This requires an input of energy, so this would be a positive delta G. All right, this is an endergonic reaction, a reaction where we're putting in energy to form bonds between molecules, making a more complex molecule. There's your energy input. Now, where do we get that energy from? The place where we usually get that energy from is from ATP, where we've stored this energy. ATP will get broken down, and we will have a negative delta G. That is an exergonic reaction. So a reaction giving up energy is going to be negative. Now you can see this is talking about, for instance, per mole, so for every 1 mole. So for every 1 mole of ATP that we break down, we get about 7.3 kilocalories per mole out of that. If we use that to drive this reaction down here, you can see that this reaction costs 3.4 kilocalories per mole. Now if we look at what this means that we have leftover. Our leftover is a delta G of negative 9.3 kilocalories. So this is extra energy. This extra energy is often given off as heat. So in fact, the heat within your body that's being generated within your body is actually an inefficiency of your body. So you're not using all of the energy that you should efficiently. So you can see here, like I was saying, if we imagine that this is a reaction between moles of ATP and moles of making glutamine, you can see that there's an inefficiency in the system because we're basically creating too much energy for breaking down 1 mole of ATP. There's leftover. This leftover is almost always heat. The place where we normally see a problem with this leftover, because normally, right, your body is 98.6 degrees. So this leftover energy that's creating heat in that case is actually a good thing because it's keeping our body at the temperature where we want it. When this becomes bad and you can really see this, is when you exercise. So if you exercise, if you've noticed, what happens is you heat up, and you heat up so much that you actually need secondary systems to help dissipate that heat in the form of sweat. This is just a couple of very inefficient systems within your body where when you're exercising, you're having leftover energy that you're not burning, and so it has to be released as heat. That heat then needs to be dissipated, and we have to create a whole nother system for dissipating that heat in the form of sweat. Okay, so that's energy. Now let's talk about enzymes. So enzymes are incredibly important in your body. What they do is they couple the release from exergonic reactions and endergonic reactions. So in other words, they help with both exer and endogonic reactions. They are primarily proteins that are going to act as enzymes. So a lot of the proteins in your body are enzymes. They basically help to move these reactions along. The enzymes act as catalysts, so that's what I mean by they help to move these reactions along. They're not consumed or altered by the reactions in the end. They can be used many times repeatedly. This is what catalysts are. And the presence, absence, and concentration of an enzyme in a cell is determined through its genetic expression. So basically, the genes that are turned on or off within an individual cell will determine what enzymes are going to be produced. And this in turn determines what the cell is going to do. And then the enzymes are regularly replaced as well because they do wear out after a while. Now, let me talk about this real quick. So you may notice, for instance, like a liver cell will do completely different things than a kidney cell. yet they share the identical and exact same DNA. And so they have the exact same genetic structure, but they do two totally, completely different things. The reason for this is because of genetic expression. So basically, certain cells will only turn on certain genes out of their entire genetic set and use those. And that's why a liver cell will act completely differently than a kidney cell, even though it has the exact same information in it in the form of DNA. So, and that's called genetic expression. All right, so it can turn on or off certain parts of its genome based on where it's located and what it wants to do. Now, this genetic expression can also be influenced, and it can be influenced by things like hormones that can come around and talk to the cell and tell it to do different things. But obviously, like a liver cell is not going to suddenly become a kidney cell. It can only be influenced to do slightly different things along its own pathway. All right, so let's talk about enzymes. Enzymes themselves, remember that they're primarily proteins. They must have an active site. And an active site is the site where something is happening, where it binds to another molecule and changes that other molecule. That other molecule is typically called a substrate. Another term that you may hear is ligand. So those are very close to being the same, a ligand and a substrate. And those have to be complementary in size and shape to the active site. So in other words, the substrate is the molecule that's going to enter the active site of the enzyme and then be changed. This can also be called a ligand as well. Okay. Induced fit means that the substrate must deform the enzyme somewhat in order to initiate this process. So there has to be a little bit of movement of the enzyme. And then the activity is concentration dependent. So it's dependent on the concentration of the substrate. So the activity, in other words, the activity of the enzyme is. If we take a look at this picture here, this picture shows an enzyme shown here in purple. Okay, you see it just has two parts of this enzyme. There's an active site shown here, and here you can see a substrate that's about to enter this active site. And in this case, the substrate is glucose. So substrate is just a generic term for anything that's going to enter the active site. You could also call it a ligand. Once this glucose enters into this active site, what happens is it causes, you can see here, kind of a crunch or a change in shape of the enzyme. And when the enzyme changes shape, it also squeezes on the substrate and causes the substrate to become a different molecule. So for instance, here we have glucose coming in. What's coming out is a different form of glucose. All right, so this is not going to be glucose anymore, but it might be something like glucose 1 alpha or something like that, right? That will pop out of here. And eventually, once this has been squeezed into shape, its new shape, it will pop out and the enzyme will return back to its original shape. Now, an enzyme doesn't have to just act on one molecule at a time. It can sometimes change things by binding to multiple substrates. So here, for instance, we have an enzyme. We have two substrates that have to enter into the active site here. So this requires two different molecules to enter. You can see the two molecules will enter, and when they leave, they come out as different products. So you can see the original substrate A has had a portion of itself cleaved off, and it is now what we call product C. And you can see substrate B that originally entered here has now had an additional part added onto it that was moved over from the original substrate A, and it is now product D. And so you can see this drawn out here. This is commonly how we draw this. We have the reactants on one side, they enter the enzyme. Usually we just have an arrow here that indicates an enzyme, and then we have two different products that are coming out. Now, we can have ways of influencing these enzymes as well. For instance, there can be what are called allosteric inhibitors or allosteric activators. All right, so let me draw this out here. And let me clear off our old stuff here. All right, so this is going to be different ways that we can activate or deactivate enzymes. So one thing that your body does not want to do is it does not want to always have all of its enzymes running all the time. So it needs ways to activate or deactivate the enzymes. Here are several ways that it accomplishes this. The first is called proteolytic. So proteolytic, these are going to be activators, okay? So I'm going to start with the activators. In proteolytic activation, we have a protein that's been built, which we would call like a pre-pro protein. And this protein has been built, I'm just going to draw it like this with a couple of parts in red. So imagine that this is a protein chain, and I'm putting in two parts that are red in color here. This is an inactive protein. Okay, so no activity on this protein right now. What we could do is we could cleave this protein and pull out these little red chunks. So here we pull out the little red parts, and we're going to put them over to the side over here. And we'll take the rest of this and we'll form it together, and you can see that it forms a slightly shorter chain now. This is now an active protein, okay, or an active enzyme. This is proteolytic activation. So in other words, you build the enzyme, but it has these parts in it that make it inactive. And then when you want it to become active, you cleave portions of it out. So basically this is peptide cleavage. Another way to activate an enzyme is through what's called an allosteric activator. In an allosteric activator, we have an enzyme, let's say it looks like this. On the back side of it, it has this little bump. And right now, this enzyme is inactive. And imagine that on the green area over here is the active site that is not active right now, okay? We can have another molecule that comes in, and let's say it fits and can move in and fit into this space. And so this molecule now attaches into this space and fills this site like this, okay? When this happens, this now changes this molecule on the other side and causes it to now look like this. So here, I'll draw this again on this back side. And now on the other side, we might have the opening of the active site. So here now we have the allosteric activator actively attached into this site. And now this molecule is now active. And you can see here the active site is now open. This is called allosteric activation. So allosteric meaning away from the active site. So we have something acting on a different area of the molecule. All right, another way to activate these enzymes is through what's called a cofactor. And cofactor activation is very similar to allosteric activation, but here these are typically going to be like individual ions or metals. So metallic ions are often what they are, that are used to activate the enzyme. They attach to an enzyme to activate it. So you might have something similar here with an inactive enzyme. And then when you add magnesium to it, so magnesium, I can't remember if it's 1 plus or 2, it doesn't matter. When magnesium attaches to it, that again opens up the active site here. So, but here the magnesium is actively attached to it. That's going to be a cofactor. Usually cofactors are non-organic type of materials. Other examples would include zinc. So there's a bunch of metals that can be used for this. Hang on, let me get there you go. So Things like zinc, iron is another one. Calcium can sometimes be used for this. Magnesium, which, oh yeah, it does have 2 pluses on it, and potassium also. Any of those can be used as cofactors. Okay, so those are activators. Now let's take a look at inhibitors. So inhibitors are very similar. Let me clear this. So first of all, for inhibitors, we'll start with allosteric inhibitors. This is the exact same concept as allosteric activators, except here what's happening is the enzyme has an open active site. So the active site on this is active, so this is going to be an active enzyme. And then when another molecule that fits the shape on the back here comes in and locks into here, it basically turns this off. So it's the same thing as before, except now you're turning off the enzyme instead of turning it on. So here now we have the allosteric inhibitor locked into the back of this, and here we've turned off the active site. So this would be an inactive molecule. There you go. All right, and then the last type of inhibitor here is what's called a competitive inhibitor. A competitive inhibitor actually binds to the active site itself. So imagine that we have this active site like this. This active site, this is currently an active molecule. A competitive inhibitor will be built in such a way that it takes up all or part of the active site. So here I'll do one that's just part of the active site. Imagine it looks like this. It can come in and insert itself in here, and when that happens, it basically blocks the active site so that other molecules cannot use that active site, right? So now it's actively blocking that. So this is a competitive inhibitor here. It's competing for the active site. All right. So now let's take a look at other influencers on enzyme activity. And there are a number of things that can influence enzyme activity, and the reason why I'm spending all this time on enzymes, by the way, is because they're extremely important in physiology and biochemistry. So here you can see that enzyme activity is affected by temperature and pH. So these are two of the biggest factors that will affect enzymes. Temperatures that are too high will denature an enzyme. So denaturing means unraveling. They only, though, unravel the secondary, tertiary, and quaternary structures. So these numbers are referring to the structure of the enzyme. So secondary, tertiary, and quaternary. They do not denature the primary structure. The primary structure is the connection between the amino acids. So in other words, even if temperatures get very high, typically amino acids themselves do not come apart. The chain stays together, but all the other structures within the protein fall apart as temperatures get higher. The other thing that can affect this is pH. Now, pH is a little more variable in that depending on if the pH is high or low, and depending on the enzyme, this will affect whether or not it destroys the functionality of an enzyme. So, for instance, in us, let's use humans as an example, we have a steady temperature of around 98.6 degrees Fahrenheit, right? So our enzymes all work at the same temperature, right around 98.6 degrees. However, within the human body, we have areas that have high pH and areas that have low pH. So the enzymes in those areas have to be specific to those pH levels. So for instance, within your stomach, we have very low pH because there's a lot of hydrogen in your stomach. This can easily denature an enzyme that's not accustomed to this low pH type of environment. So the enzymes within your stomach, so like pepsin, let's say, are actually engineered in such a way that they work in low pH environments. Now, right next to that is an area that has a very high pH, which is in your small intestine. So the enzymes in your small intestine have to be specifically built to work at high pHs. So you can see how in the human body, because the temperature is the same everywhere, all your enzymes work best or optimally at the same temperature, but the pH is different in different parts of your body. So because of that, the enzymes have to be tailored for that specific pH. And this is true of all creatures. This is not just us, even all the way down to bacteria as well. Now, other things that can affect enzyme activity, I talked about the availability of cofactors. Prosthetic groups are something very similar. You don't really need to know about those, but they're basically a type of cofactor that binds very tightly to the enzyme. That's a prosthetic group. Same thing as a cofactor, essentially. Competitive inhibitors, like I said, that will bind to and take up the active sites. And non-competitive inhibitors that affect the shape of the active site. So a non-competitive inhibitor, which is one that I did not really discuss, is basically it's the same thing as an allosteric inhibitor. It's just that one is a subtype of the other. So a non-competitive inhibitor is a type of enzyme inhibition where the inhibitor reduces the activity in the enzyme and binds equally well to the enzyme whether or not it is already bound to the substrate. So it sounds complicated probably, but basically what I'm talking about here in this non-competitive inhibitor is an allosteric inhibitor. That's what we're really looking at. All right, this picture just shows, it's kind of interesting because it shows what I just talked about how enzymes can react differently depending on the temperature or the pH that they're put in. So let's talk down here first on this bottom picture. It shows the optimal pH for pepsin, which I mentioned is a enzyme in your stomach. Notice that the optimal pH for pepsin is 2. And that makes sense because your stomach has a lot of acid in it, which means a low pH. And so it works optimally at low pHs. But now look over here at trypsin, which is another enzyme. This is an enzyme found in your small intestine. Your small intestine has a tendency to be alkaline or basic, and you can see here that trypsin works best at a pH of 8, which is alkaline. And so you can see here how even though these two enzymes are both within the human body, they work at very different pHs, and if you put one in place of the other, it wouldn't function because the pH of the solution would destroy the functionality of that enzyme. So in other words, if you took pepsin and stuck it into the small intestine, it's not going to work or work at a very low level. And if you took trypsin and threw it into the stomach, it would not work or work at a very reduced level. Now, that's compared to temperature. Remember how I said all of our enzymes work in optimal temperature around 98.6, which is 38 degrees Celsius, which is shown here. So all of our enzymes in our body all work at this optimal temperature. But here are enzymes within heat-tolerant bacteria that have been found in places like, what is it, Yellowstone? Yeah, Yellowstone National Park, where they have all the hot water geysers and things like that. The bacteria in those areas are actually very heat tolerant. And what they found, researchers have found, is that within these bacteria, they have enzymes that are actually most active at around 75 or 80 degrees Celsius. So if you took those bacterial enzymes and tried to put them into us, they wouldn't work. If you took any of our enzymes and put them into the bacteria, they won't work if they're in their normal environment of either cold or hot, right? So again, I'm just trying to emphasize how enzymes can be affected by both temperature and pH, and they can lose their functionality if they're not in their happy zone, basically. This just shows the pH effect on different digestive enzymes shown here. Here we've already seen pepsin and trypsin, but you can also see how salivary amylase has an optimal pH of around 7, which kind of makes sense because it's active in our mouths. So these slides here just show more pictures like I just drew of competitive inhibitors that are taking up the active site and therefore they block the substrate from getting in. And then a non-competitive inhibitor is shown here where it binds to an area other than the active site, so not on the active site, and it actually closes the active site to keep the substrate from getting in to this enzyme. These are all pictures that I've drawn for you, so I'm just basically reiterating it with the drawings from the book. Okay, so control of metabolism. So remember how I said that enzymes, your body does not want all of its enzymes active all at the same time. So it has ways of either inhibiting them or ramping them up depending on what it wants to happen. So control is maintained oftentimes through these allosteric inhibitors. And these allosteric inhibitors are often part of a pathway, and they feed back on themselves in order to control the pathway. Okay, and I'll have a picture coming up here that will show you this better, because I know it's kind of hard to understand in words. The controlling factor is the concentration of a substance related to the pathway, and this allows for alternate use of the metabolic intermediates as well. So let me show you this as a pathway. This is very, very typical of how your body works, this pathway shown here. Okay, so here you can see that what we have is threonine, and we want to turn threonine into isoleucine. So that's our final product that we want to get it to. This is very typical of how your body works. It's often not a one-step reaction. Instead, you can see here that what happens is we have an enzyme called threonine deaminase in this case that turns threonine into an intermediate that we're not drawing here or showing. This intermediate A will be reacted on by a second enzyme that will turn it into intermediate B. That will be reacted on by another enzyme that will turn it into C, enzyme 4, D, enzyme 5. We finally get our final product. This is very typical of a biochemical pathway within your body. All right. Now, how do we turn this system off if we have enough isoleucine created? Well, it turns out that the way that your body does this, it'll take the end product, which is isoleucine. This will circulate back around, and it will actually turn off the first enzyme. So threonine deaminase is sensitive to isoleucine. So isoleucine may block its active site. It might bind to threonine deaminase in such a way that it is allosterically turning it off, but in some way it's going to inhibit this enzyme. Now, what that means then is that we have a buildup of threonine if we've turned off the production of isoleucine. So now threonine can go and follow another pathway if we need to get rid of some of the threonine, or if we need other substrates built up. But this is very typical of how your body works. It takes the final product or one of the products in the pathway and uses it to turn off one of the early enzymes in the pathway. This is also shown here. So here you can actually see an alternate pathway. So imagine here that we start with a reactant A, and enzyme 1 changes it into B. Then we have another enzyme changes that into C. Enzyme 3 changes this into D, enzyme 4 into E, and so forth down to F. The final product, F in this case, does not go back and turn off enzyme number 1, but instead F turns off enzyme number 3. So it has a negative feedback on enzyme number 3. And again, this is a very common thing that happens. It doesn't have to necessarily turn off the first enzyme. This turns off as F builds up, so F prime down here, builds up. It will eventually turn off enzyme 3, either allosterically or by binding to its active site. This in turn leads to a buildup of product C. As product C builds up, now enzyme 3 up here, sorry, this was 3 prime down here. Enzyme 3 up here takes over and turns this into D, which then turns this into E, and this enzyme turns it into F. This is a secondary pathway that now we have product A forming and becoming eventually F instead of F prime. So this is, like I say, how your body works all the time. Here are examples of this. So here's an actual real example of it. Here's phenylalanine. This can be used to make melanin. But once we get enough melanin built up, we can then use the secondary pathway to just metabolize it down into carbon dioxide and water. So basically, if we have enough phenylalanine and we have enough melanin, and we can also turn it into phenylpyruvic acid, if we have enough of all of these, then the final pathway can basically just turn it into a metabolite that we can get rid of. So basically, that is how these pathways work. And down here you can see some defects in the body. So I always like to talk about some of the physiological problems that can arise with whatever I'm talking about. So in this case, these are metabolic defects that can happen. and they result in specific diseases. So for instance, here you can see a set of amino acid metabolism diseases. So fetal ketonuria is one, albinism is another, and then there's another one called maple syrup disease, kind of interesting. You can see what the abnormality is in each of these and the clinical results. So we'll talk about a number of these diseases throughout the semester, but it's just kind of interesting how you can have these metabolic diseases that are basically because of these pathways not functioning correctly or specific enzymes in the pathway not functioning correctly. So with that, that is the end of my lecture for today, and I hope you have a good day. Thank you.